Hello guys!
Today we're going to talk about cytoskeleton
and Centrioles.
I'm starting our presentation with the cytoskeleton. So, the cytoskeleton is responsible for the cell shape, movement of the cell as a whole and the movement of organelles within a cell. There are three types of filaments in the cytoplasm of most vertebrate cells: microfilaments, microtubules and intermediate filaments. All of these filament systems share a critical feature. They are composed of proteins that have the unique property of being able to self-assemble into a filamentous network. So, imagine a pile of bricks that could assemble by themselves into a wall. The proteins that make up the fibers of the cytoskeleton are able to just this. The proteins that make each of the three different filament systems assemble into the only structural characteristic of that filament. Unlike the human skeleton, the cytoskeleton is extremely dynamic, meaning the filament systems are able to lengthen or shorten very rapidly. This dynamic nature of the cytoskeleton is necessary for the cells to be able to change shape, complete cell division, or migrate, and represents one of the cytoskeleton's most important features. Each of the self-assembling proteins has a characteristic concentration called the critical concentration, below which the monomer state is favored and above which the polymer state is favored. Increasingly, the submit concentration favors a filament building and decreasing it into favors filament deconstruction. This property allows the cell to rapidly control cytoskeletal structure. Talk about the microfilaments. The microfilament system is a network of filaments which is in length 6 nanometers in diameter that are important for anchoring plasma membrane proteins for producing cell movement and for cell division. The base filament is composed of a protein called actin that is 42 kilodalton in weight. Actin is also a protein that forms into the thin filaments found in muscle. When purified, actin is inhibited in a test tube. 6 nanometers filamentous structures are formed. These trees consist of side-by-actin monomers that twist around each other in a helix. Inside cells, actin exists in two states: monomers that twist around each other in a helix. Inside cells, actin cells in two states: the monometous protein called G-actin for globular actin and the 6 nanometers filament called F-actin for filamentous actin. The factor that determines the proportions of F-actin and G-actin is the concentration of action protein. Each microfilament has a fast-growing or plus end and a slow-growing or minus end. In the most cells, the plus ends of the filaments are oriented toward the edge of the cell. In this way, rapid polymerization of action monomers onto the cell plus ends of microfilaments can produce protrusions of the cell surface called pseudopods. These extensions are critical for the ability of cells to migrate in directional fashion. Microfilaments exist in the highest concentration in association with the cell periphery, where they are believed to play an important role in anchoring membrane proteins. Microfilaments can also be organized into bundles called stress fibers, which serve as contractile elements somewhat like muscles within cells. These structures are important for maintaining connection between the cell and the surface on which it are important for maintaining connection between cell and surface on which it grows. In addition, these structures may be important for producing contractility into general directional force during cell motility. A third microfilament-based structure is the contractile ring. It is critical for the separation of the cell into two progeny during cytokinesis. As you can see, here is the cell, and here are the actin filaments. Microtubules are the largest of the cytoskeletal filaments with a diameter of 25 nanometers. There are many parallels between the microfilamental cytoskeletal system and the microtubule system. Like microfilaments, microtubules are produced by the self-assembly of a subunit, which in the case of the microtubules is a heterodimer composed of one alpha tubulin and one beta tubulin bound together. Alpha and beta subunits alternate to form a protofilament. 13 protofilaments line up and side by side, forming the hollow tube of the microtubule. Microtubules also have a fast growing or plus end and a slow growing or minus end. In most cells, microtubules are organized in a radial array extending from a single side, termed the microtubule organizing enter MTOC, generally positioned near the nucleus. This organization produces a network of microtubule tracks where the plus ends of the microtubules are near the cell surface and the minus ends are associated with the MTOC. This structure is well suited for the primary function of the microtubules, which is to serve tracks along the membrane-bound vessels are microtubules. Vesicles transported include organelles such as mitochondria as well as secretory vesicles detained for exostosis. Another parallel with the microfilaments is the highly dynamic nature of microtubules. Microtubules exhibit a phenomenon called dynamic instability. Individual microtubules constantly grow and shorten, often shortening dynamically in a process called catastrophe. The rapid turnover of the microtubules allows cells to change shape quickly and facilitates the reorganization of the tracks important for the delivery of vesicles to the sites throughout the cell. Like the microfilament skeleton, the dynamics of the microtubules can be modified by microtubule-associated proteins called MAPs. Some MAPs stabilize microtubules, which others cross-link microtubules both with other microtubules as well as microfilaments, and the third cytoskeletal system, intermediate filaments. The dynamics of the microtubules are also important for the mitosis. Each time the cell goes through division of the microtubule, the network is completely disassembled and the tubulin subunits are assembled into a new structure called the spindle. The spindle is responsible for the segregation of chromosomes into each daughter cell and also plays an important role in specifying the position of the cleavage plane that will separate the two daughter cells during cytokinesis. The functions of the microtubules in the vesicle transport and chromosome segregation are dependent on molecular motors that bind to move along microtubule tracks. These motors are divided into two families, kinesin and cytoplasmic DNA. Kinesin was the first microtubule motor to be identified. It is responsible for moving vesicles, the cargo of the motor, toward the plus ends of the microtubules, that is, from the center of the cell toward the plasma membranes. Since discovery of the first kinesin, the family has been shown to consist of many members, some of which are important for the spindle function during mitosis. Some of these kinesins move toward to the minus ends of the microtubules. In contrast, the other type of microtubule motor, cytoplasmic DNA, appears to move cargo exclusively toward the minus ends of the microtubules, that is, from the cell periphery back towards the center. The minus ends of microtubules, that is, from the cell periphery back towards the center. The ability of these motors to move organelles around inside cells is critical for the processes such as hormone secretion, transmission of nerve impulses, and recycling of membrane. As you can see, here is the cell. This is the cell nucleus, which is the black thing. Here are the actin filaments, which are located on the edge of the cell. You can see them here. The centrosome is located just over the cell nucleus, and from the centrosome are coming these microtubules, which I was talking about them, and they're like the cats, these things. Here are the intermediate filaments, these things in gray. So they are coming out of the actin filaments, they are actually connected with them. As you can see, this is another image of the cell, how it looks like. So this is the mitochondrion. It looks like, it looks very strange. Here are the microtubules, which are located just here, if you can see them like sticks. Here is the intermediate filament, which is located just here. It is connected to the actin filaments, and the actin filaments are actually located here in the bottom of the cell. and the DNA is just located over these things. We unfortunately cannot see it.
Hello. Now I'm gonna tell you about the centrioles. Okay, so all animal cells have two small organelles, and they are called centrioles. Their function is to help the cell to divide into like two cells. The centrioles are barrel-shaped, and they are located only in the animal cells. They are located near the nucleus. Each centriole is made out of 9 bundles of 3 microtubules. Each microtubule's diameter is around 25 nanometers. So, in each cell there are 2 centrioles, and they are arranged in a perpendicular angle like that, and they are stuck together. Also, they appear only, usually they are not in the cell, but during the mitosis, the dividing cell process, they appear and they help this process. I changed the location because there was too dark, and here is better because of this lamp. Beautiful lamp. Okay, so now I'm gonna tell you about the function of the centrioles. The centrioles are located only in the animal cells, and because the plant cells don't need them, they can divide without them. So the centrioles in the animal cell are involved in 2 main processes of division. The centriole, the process of mitosis, and the one of meiosis. So the centriole's function as the microtubule organizing center is important element in a major cellular process. So this main process is the cell division. So the process, like the function here in this process of the centriole, is to show where the nucleus of the newest cell will be. So the process is like that. The 2 centrioles go on the opposite side of the cell, and they form from there they form like 2 new centrioles that are attached to each other, and these 2 centrioles determine the position of the nucleus in the new cell. Also, the location of the new centrioles influence of the location of the other elements of the cell. So also, after this process, in each cell there are again two centrioles that will be useful in the next process of division.
That was from us, the grammar professors. Thank you for your attention.
Thank you!
Bye.